Well, first, I'd like to thank the organizers for inviting me here to share the data with you. And I'd like to start with this. As a junior faculty, I started my lab at Duke University. I was so stressed out, I didn't know what to do. And I was thinking, if you are doing research, you want to do the forefront research, you are almost like a fly in the space, and you only have enough fuel to come back to Earth. And so which direction you want to go? You want to go fly fast, or you want to find the right direction? I think the second answer is the correct one. I would ask David, will you agree with me the center of the universe is this? So mitochondria is present in literally all cells. Probably erythrocyte at the end stage that got rid of mitochondria. But mitochondria is the organelle in our body that is where ATP is generated. So ATP generation is the main function of mitochondria, but mitochondria is also a sensor of energetic stress or status. So therefore, it becomes fundamentally important when mitochondria is damaged. If it's not causal, at least exacerbating many, many conditions. The cell can sense it. So the question is, what's the sensor for that? So the reducing equivalency go through the electron transport chain will be used to generate, do I have the laser? To generate ATP. If the mitochondria is leaky and the small molecules change in membrane potential, everything can elicit signal for cells to say, okay, we got to stop. And we have a problem with the power plants, and then we have to stop doing the normal function. So many examples showing that mitochondrial dysfunction is tightly associated with a disease, such as type 2 diabetes. The second one is heart failure. Before you can detect a functional decline in the cardiac function, you start to see abnormal mitochondrial structure and function. And even in the autopsy samples from the AD patients, you'll see damaged DNA in 4 different regions of the brain, either nuclear DNA or mitochondrial DNA. If not more, mitochondrial DNA is more prevalent there. So, but positive side, my very good friend, John Hallosi, many years ago, the grandfather of exercise biochemistry, which, the name of the meeting, and show that exercise is really the powerful intervention to enhance mitochondrial function. In this table, you can see after a short training period, in either, this is the rat study showing increased mitochondrial enzymatic activity in gastrocnemius and the soleus muscle. So I miss this guy a lot. So that was my decision. Go for mitochondrial work. So follow the mitochondrial work. And over the years, I focus on the importance of PGC1 in coordinating the nuclear genome and the mitochondrial genome in making new mitochondria. So maybe I found this. So making new mitochondria. So for the first 10 years, I spent on studying PGC1 in control of mitochondrial biogenesis, inspired by Bruce Spiegelman. So Bruce and I collaborated since 2002, so 20 years already. And we studied with these many papers published, we show that P38 gamma and through the transcriptional control of PGC1, and we confirmed that PGC1 is very important for angiogenesis and the mitochondrial biogenesis. But in the knockout background, PGC1 seems to be important for fiber type switching. When you overexpress PGC1, you can have change in fiber type to a certain degree. But when you get rid of PGC1, you cannot affect the transformation of fibers. Therefore, PGC1 is essential for metabolic adaptation, not necessarily for contractile adaptation. Now, one of the technique I'm proud of is this anecdotal thing. I was walking the hallway. I said, I'm studying PGC1. I'd like to know PGC1 promoter element that is important for PGC1 function. Somebody said, do you know you can use luciferase in vivo? I said, how could you do that? And, but indeed, the technique I learned with Dr. Frank Booth by doing electroporation, we did that with the contralateral leg and with injection of luciferase with the PGC1 promoter, and then you can measure that after pace the muscle. So you can use the contralateral as control and use the ratio to see what happened after 10 days after transfection. You pace it for an hour. You can see the increase in the PGC1 luciferase, but not in the RSV luciferase. You can mutate different sites. So by doing that, we were the first one to really use PGC1 promoter to study transcriptional control in vivo rather than in the tissue culture dish. So, and then 2006, I started to switch to study the other side of the coin, mitophagy. Mitophagy, the first speaker, Andy, really gave a great review, and so I don't have to go through that. And it's a very coordinated event leading to the removal of damaged mitochondria. So this is really, we are excited to see that a high-fat diet causes accumulation of damaged mitochondria, but with the exercise training, you start to see the double membrane engulfing the mitochondria and some even with the remnant mitochondria structure there. So at the time, autophagy was a new term. And so that was a direction thing. We have to decide whether we want to pursue this. So I made a commitment. I said, I pursue this. I may be very slow, but I'm doing it. Rihanna, trained by Australian group in Melbourne, another anecdotal story, I went to Melbourne for that 2000 meeting. And then Rihanna approached me, said she wanted to pursue a postdoc career with me. So when she came, I told her that, we have a hard time to detect mitophagy using EM. It will, cost years of life to do that. And a friend of mine from Singapore, who is the director of the Singapore Imaging Consortium, he said, do you know that there is a timer that you can use to measure oxidative stress? So I said, okay, if I targeted to mitochondrial matrix, using this mitochondrial targeting sequence through the TOM and the TIM, getting to the mitochondria, we may be able to measure that. Again, electroporation, beautiful structure shown, and with the punctate. So Andy showed that data, and we confirmed it's a LAMP1 positive. Now with more than 500 labs sharing this reagent, we made tissue-specific transgenic animal reporter gene now. So with that, we started to address the question whether acute exercise will promote mitophagy. Our data show that even with a single bout of 90 minutes treadmill running, we don't see a change in color until 6 hours later with accumulation of red dots, which is the indicative of mitophagy. And with a contralateral leg, this is another thing for the trainees. It's important to do the control. So we use the timer, but targeted to ER to inject into the contralateral leg. So this control is a contralateral leg. We injected the ER timer. They didn't show any change in color or appearance of any red dots. So that validates this data is real. So Josh and Rihanna really collaborated beautifully on this project. So then we asked the question, what is the thing turn on this? If there is kind of a oxidative stress to mitochondria, mitophagy, what is the regulator for the clearance part? So if you can imagine, like there's a fire, somebody called a 911, and somebody have to come to the rescue, right? So this is the question. Where is the headquarters? So we believe that Ruben Shell's data inspired us to look into the importance of AMPK. Because if you think about all kinases, AMPK is the energetic sensor. Mitochondria is the ATP production site. So we asked the question whether indeed OAK1 serum 5.5 is important for that, AMPK is important for that. So in his paper, he showed that if you knock out of AMPK, you have accumulation of mitochondria. If you knock out of OAK1, you have accumulation of mitochondria, indicating AMPK OAK1 pathway is important. So we collaborate, and so that was 2018. I came to Niagara Falls, and first time I met Graham Hardy in person. So I talked to him about the project on the bus to the fall. And I said, I want to invite you to UVA. And he came, and then we started to collaborate. So AMPK is the energetic sensor with 3 subunits, 12 different ways in combination. But the gamma subunit is where ATP, ADP, AMP can bind. And the binding, particularly of AMP or ADP, can trigger the transformational change, expose the alpha subunit to the upstream kinase. So if you think this is like a 2-step of activation, binding, allosteric binding of metabolite and lead to the phosphorylation post-translational modification, lead to a full activation. Binding of ADP or AMP can turn on AMPK, increase the activity by 5-fold, 10-fold, but with the kinase, it can go 100-fold. So the first question, Josh, who is now a professor, assistant professor with tenure track with a great funding at Virginia Tech, and asked was to detect AMPK phosphorylation in response to 90 minutes running, and to see what goes with it. So indeed, if ACC phosphorylation increased here, a very classical AMPK substrate. But here, the OAK1 phosphorylation increased concurrent with AMPK, but not the inhibitory phosphorylation side by mTOR, serine 757, and the serine 467. So total OAK1 did not change, total AMPK did not change. So with that, we collaborated with Lori Goodyear, and we got the dominant and negative AMPK transgenic animal, specifically in skeletal muscle, show that an acute exercise-induced serum 5.5 phosphorylation is blunted. And then when we use the constitutively active AMPK transgenic, we saw increase in ACC phosphorylation and OAK1 phosphorylation. So Josh confirmed AMPK is sufficient and necessary to induce OAK1 phosphorylation. Then he did the co-transfection of the mitotimer with the reported transfected mitotimer into either the transgenic animal of dominant negative AMPK or muscle-specific knockout of OAK1. And basically, he showed the same thing, that exercise-induced mitophagy, as indicated by the puncta with the LAMP1 positive dots, and blunted in the either muscle-specific OAK1 knockout or dominant negative AMPK transgenic animal. So, but that data really gave us this working hypothesis that exercise can promote stress to mitochondria, maybe even damage the mitochondria, which is not optimal. But over the years, you have aging, you have toxins. Some mitochondria are already partially damaged, just like the car, the damaged car clunkers. And then exercise can further trigger the stress to those part of the mitochondrial network. At the same time, AMPK is activated and phosphorylated OAK1. OAK1 can promote this part of the autophagy pathway, but we also show that OAK1 actually promote the fusion of this autophagosome with a lysosome. So that's the working model we had. Now the question is that muscle is full of contractile proteins. If you imagine that after a game celebration, your buddy is over there, you try to find them, it will take forever to go there and find them, right? So that doesn't not sound right for a biological system. So Josh and I talked about it. We said, what if AMPK is close to mitochondria or physically associated with the mitochondria? So he did this work with isolation of mitochondria with the Perkel gradient, the gold standard, and he saw AMPK. And then he confirmed is the AMPK alpha 1, alpha 2, beta 2, and the gamma 1 in the mitochondrial fraction. And then he did the peptide blocking to confirm the identity of those bands. But one experiment he did was most beautiful one, was that to gently digest the isolated mitochondria here with the trypsin, leaving the COX-4 intact, but the rest of that disappeared. So that indicates that AMPK is on the outer membrane of mitochondria, recruited by something we don't know. And then we collaborated with Graham Hardy to show that the whole cell lysis, cytosolic fraction, mitochondrial fraction, they all had mitochondrial activity, and with addition of AMP, you can double the activity. So we know it is present. Then the question, is it activatable? So we did the treadmill running. We saw increasing AMPK, mitoAMPK phosphorylation, but not in the dominant negative transgenic animal. So there are two things. Dominant negative is an overexpression of a non-activatable AMPK, so you can block that by doing that. And then the kidney, 5 minutes of ischemia, you see this activation of mitoAMPK. This is a mitochondrial fraction AMPK. No change in total AMPK. Then the most important one, I think, is the metformin. We all know metformin is an anti-diabetic drug. And with the injection of metformin, you can activate a mitochondrial fraction of AMPK, have very little impact on the cytosolic AMPK. So this really gave us the indication that either way, you can push the pedal, increase the engine to stress the engine. You can also inhibit the engine, block the engine from running, but the engine will try to run to stress the engine. So both ways can activate the mitoAMPK. Another approach we used is the Fred imaging. We use this reporter with 2-fluorophore with the phosphorylation site for AMPK kinase, not the phosphorylation site on AMPK, but the substrate, and then target it to outer membrane. Then you can see this, although the resolution is lower, you can still see the structure of the mitochondria. And that's the stimulation. 20 minutes later, you see the activation of mitoAMPK. Even with the tissue culture with the oligomyosin inhibition, within 15 minutes, you can activate a mitoAMPK. And the most important question is that, is it functionally important? And so we used the same approach that published in 2016 by a group over in Johns Hopkins. They used the peptide with the AMPK motif, same approach, overexpress that, and then you can see this accumulation of mitochondrial content, but not with the mutation of threonine to alanine. And so even though it's the same construct, but lost this phosphorylation site, then we lost this effect of accumulation. So this shows that if you inhibit the mitoAMPK, you can block mitophagy, similar to Rubin Shell's data in the hepatocytes. Finally, we co-transfected this we call it mitoAIP. It's a mitochondrial inhibitory peptide with the mitotimer. You can see this expression of that caused some stress, but exercise-induced stress, you can see that there is a trend here. Here is the pure red puncta or autophagy. You can see the increased autophagy significantly reduced in the AIP transfected fibers. So at the time, we say, okay, AMPK phosphorylation is important, mitoAMPK is important. I talked to Lori, I said, has anyone really tested importance of threonine 172 phosphorylation? She said no. And we have ability to do CRISPR-Cas9. So we did it. I talked to Lori. When she said green light, I moved. And so we made all these knock-in mice. Now I'm going to give you the example of alpha-2 knock-in. You can see this is the knock-in mice. knocking mice, wild-type knocking. So all the knocking mice lost the phosphorylation of AMPK because the alpha-2 is the dominant form in skeletal muscle. And you can see this ACC phosphorylation reduced, the ORC1 phosphorylation reduced. So we confirmed ACC and the ORC1 are downstream effector of AMPK alpha-2. And then you can see there's a reduction of COX-4 cytochrome C. There's a little bit reduction of mitochondrial function. And then we put those mice out, and we made those mice not in black 6 background. It took us 3 years to breed that into black 6 background. And we saw slight reduced body weight, slight increase in the fat mass, nocturnal RER, not really fundamentally different. Activity is reduced. So this is the locomotive activity. There are even slightly improved glucose tolerance test result. But most strikingly, you put them on the treadmill, you do a treadmill running, they only run 1/3 of the distance, more increase in lactate for the running, put them in the metabolic treadmill, and you can see this distance is reduced, speed reduced, VO2 max reduced, here, RER, shoot to the roof. So there is a major problem probably with mitochondria or regulation of fatty acid metabolism. So they cannot use the fat to sustain the activity. So that is the data with the reduced the fatty acid oxidation, increase the carbohydrate metabolism, anaerobic threshold reduced. So these mice are problematic, and we are at the very beginning to characterize that, but we put a lot of effort. You see the number of animals here. Finally, we decided to take the same approach. We got challenged by many that, how do you know that AMPK knock-in targeted mitoAMPK, right? So we know that construct is working, so we did that with a tissue-specific inducible transgenic animal. So this is a cat reporter gene with a stop codon before this AIP. And when we have no CRE, no expression, when we have CRE, we have mitochondrial structure, cell induced. Then we decide to move to make a transgenic animal. Now we have 3 lines of transgenic animal. Yun Tian is my student who asked the question, and he did this beautiful work with cardiac-specific CRE. And now this whole heart light up like the stars in the sky. And so we are very excited about elucidate the functional role of mitoAMPK. So I'm going to end this talk with a movie. So this is our muscle neuromuscular junction, action potential here will transduce a signal down to the T-tubule and trigger the release of calcium. Now calcium is important for muscle contraction. The muscle contraction need ATP, so the blue dot ATP will come from mitochondria, and ADP will go back to mitochondria for resynthesis. But when a part of mitochondria is damaged or poor in function, then the ADP will accumulate relatively. You probably won't be able to see that until unless you have a good reporter. And so now this ADP will bind to AMPK and trigger the AMPK phosphorylation activation, and the AMPK can phosphorylate other factors like MFF for DRP1 recruitment, and this DRP1 recruitment AMPK can also phosphorylate ORC1 and promote mitophagy. And this fusion of mitophagy, autophagosome with a lysosome will degrade the large content of mitochondria. So I would like to thank my lab for the work and beautiful work done by Rihanna and Josh Drake. She's with industry, and also I have a postdoc, RJ here, and Yun Tian asked a lot of good questions here. Thank you so much, and of course my collaborators, many collaborators here, and Lori Goodyear, Greg Sternberg, and Monica Driscoll, RJ Wessels, my collaborators here at UVA, and my lab, and the funding of the grants. Thank you.
Thank you, Zhen. This communication is now open for discussion. Question?
Thank you. Steffen Ravn, University of Copenhagen. Very cool new model. I was wondering, there's also Jan Wojciechowski did the inducible muscle-specific alpha 1, alpha 2 muscle knockout. And there they don't see any defects of mitochondria. And now you made this knock-in on the 172 and you see defects. So what do you think is going on between this double knockout, inducible, and?
Yeah, I think this is a perfect example. I would argue that if we want to study, let's say, the importance of a switch for turning on the light, turning on the AC for this room, we don't go smash everything and then study it, right? You will say, oh, I smashed it, AC is not working, light is not working, the room is not functional, which is perfectly fine. I think the more elegant way is to destroy the switch, and make the switch not functional, and then you say that switch is important for the AC, for the light. So that's a perfect example for that, yes.
Thank you.
So maybe the compensatory mechanism with the knockout of AMPK, which partner with many, many factors, will compensate. Yeah. So our body is very smart.
For sure, for sure.
Thank you.
Hi, Nolan Hoffman from ACU in Melbourne, Australia. So I was just going to ask, first of all, thank you for the elegant work and great talk. So you've obviously shown that AMPK has the ability to target mitochondria, and it looked like it's specifically the beta-2 isoforms. So I was just going to ask, with AMPK's known other functions for targeting other places in the cell, such as with the carbohydrate binding module and myristoylation groups which target it to membranes, do you hypothesize that it's a specific pool that's able to come to the mitochondria, or can every AMPK complex in the cell have the ability to target that?
I really don't have the answer to that question, but there are examples like a lysosome, AMPK located to lysosome play a very important role for glucose metabolism. So I think this is just the tip of the iceberg, and that's why I gave this name of the talk, location, location, location. It's very important to understand the location of important regulators. Otherwise, we just basically don't understand it and go knock it out and try to study it.
Great. And just a quick follow-up question. So do you think it's just ULK1 at the mitochondria, or is there a specific subset of those substrates sitting at the mitochondria that regulate these functions?
Oh, I think I only showed mitochondrial AMPK. So a small pool of AMPK is sitting on mitochondria. It doesn't seem to fluctuate during or in response to exercise training. With that said, my postdoc did some work with aged mice and showed even with the same number of mitochondria, you see reduced AMPK on mitochondria, suggesting that maybe aging-related decline in trainability is due to less of mitochondria there, AMPK there, so therefore they lost some responsiveness.
Thank you.
Thank you.
Thank you. Last question.
Hi, thank you very much for your great talk. Great model, I loved it. Just a question about maybe about location. Did you see when you exercise the animal a difference between the subsarcolemmal pool and the intramyofibrillary pool? Like, do you see when mitophagy is activated, is that affecting a compartment more than the other?
No, we have not looked at that. Those are all great questions. And we are busy with figuring out what to do with this. At this point, we don't. Yeah.
And just another question about the adaptation to a training. Do you see like more AMPK to the mitochondria after training, or do you see any adaptation?
No, we have not. We have not looked at that yet. Yeah, we have not looked at that yet. Thank you.
So much.
All great questions. Thank you.
Thank you. Thank you, Zane.